Hi there. So this chapter is going to be continuation of what we started in the last module when we looked at the urinary system. This week we shift our focus to look at the consequences of it, if you wish. Our interest will be on how the water balance, the balance of electrolytes, and acid-base balance is regulated within the body. So this is going to be much more about functional aspect than anatomical approach. If you compare it to how our previous chapters have been built up in the earlier modules. So why don't we get started with this? So when I was thinking of how to tie in the clinical relevance to this chapter, the obvious path for me was to mention that understanding the processes of fluid, electrolyte, and acid-base balance is vital when we look at the lab test results that evaluate these values. So here I have an example of what we could do to look at the fluid balance in an individual. And of course, the electrolytes and their reference ranges, for which we have lab tests too. And finally, but not least by any means, acidosis and alkalosis, when the pH values are off the scale. So really having a basic understanding of this is going to be vital, not only for understanding the goal or goals of the urinary system, but also to understand the maintenance of the balance of these in the healthy individual, so that we can appreciate what is going on when something is not within the normal values. And I think that a good place for us to start is to go all the way back to our introductory bio classes, where you would have discussed about this. Water is essential for life. Life evolved in water. Most living organisms are made largely of water. And of course, when we go to the space explorations, we are looking for signs of water as an indication that there may be or may have been life on other planets. So that's why water is so important for us. Let's have a look at how much of you is made of water. So it is going to be typically somewhere between 60 to 75% in an adult human. And there is some variation depending on different factors. For example, younger children have a little more water in their body. This is because there is relatively little body fat in them. And it actually turns out that the fat tissue does not contain as much water as other tissue types. So this also explains partially why females tend to have a little less water in their body than males. Females have a little higher body fat content, while males tend to have a little more skeletal muscle. And it turns out that there is a fair bit of water in muscle tissue, and this is actually something that I have been studying in terms of the cardiac muscles. We also see that older individuals have less water content in their body. Again, think of the reasons why this may be the case, and it makes sense. So next, let's have a look at how this water content is divided in your body. So to get a fair middle value, let's say that about 60% of your body is made of fluid. This would make an average adult about 40 liters. And this water will serve many important purposes, as you might remember from your introductory biology courses. For example, it is a great solvent, acts as a transport medium, plays a role in metabolic reactions. But what we want to look at here is that where the water is there. So we see here a diagram of a bunch of cells in one tissue. And let's have a look of where the fluid can be located. We have a large two groups. About 2/3 of all fluid is located inside the cells. So this is known as intracellular fluids. And the remaining 1/3 of all fluids is outside the cells, known as extracellular fluids. And most of the extracellular fluid is interstitial fluid. So this would be the fluid between the cells. And some other fluids like cerebrospinal fluid, humerus of the eye, synovial fluid, serous fluid, and gastrointestinal secretions are considered as part of the interstitial fluid, but the amounts of these are so small that I do not expect you to know this. And the rest is then our plasma in the blood and our lymph, so the fluid that has leaked out from the vasculature. So this is the basic distribution of the fluids in the body. And I want to share a clinical example of the importance of this as it relates to my work in the medical research. However, I would not expect you to memorize the following for the exam. I simply want to explain why it matters where the water is located at. Okay? So, what we see here is a typical way of looking at the heart with an MRI. And here we see a short axis view of the heart. Remember, the heart was cut in slices that were aligned according to the axis of the heart itself, not rest of the body. And general body planes as we know them. So I guess that this would be an oblique slice in that system. And what is noteworthy here is what MRI actually tells us. Well, MRI shows where there is a lot of fluid present. And fluid shows us up as bright, while solid tissues show up as dark. And here we have a picture that has been enhanced with gadolinium. It is simply a contrast agent that we use. But what is important is that we see this bright spot on the wall of the left ventricle. See, it has been also pointed with an arrow. And what this bright spot tells us is that this area of the myocardium has gotten damaged and it is dead. So because of a lack of oxygen blood supply during MI, myocardial infarction, this tissue did not survive. So the big question is now, is it worth doing an interventional procedure to try to revascularize the vessel that supplied this part of the heart wall? This is because doing an interventional procedure to try to revascularize is always risky. So if all of the heart wall is dead, that will be dead. There is nothing to save. So doing this procedure puts the patient only at a greater risk. But if there is actually some heart muscle to save, by doing this revascularization, we definitely want to do it. But how can we know if there is some? And this is where my work steps in. These are two novel mapping methods known as T1 and T2 maps, which allow us to see this bright area shown with arrows on both. But this area is not actually what is dead, but it also may be what may die if there is no revascularization. So by taking the area that we know to be dead from picture A and comparing it to what we see on B and C, we can see how much there is to save. So we can weigh in the risks and benefits. So that's what my work aims to do. And interestingly enough, what allows us to see this has all to do with the very topic that we have been looking at, where the water is in these tissues. So in MI, the extracellular fluid concentration increases. So there is more water outside the cells, in the space between the cells. And this is an interesting thing, and we can use it for clinical application, such as this. So, I hope that this little story allowed you to become at least a little more convinced that there is this topic that matters. It has some very direct uses in a clinical work. Of course, what I do is still research, not standard clinical practice, but I thought that it could be nice for you to see what it is that I actually do. Now, let's have a look at some of the characteristics of the water. And this should be familiar to you, revision from the introductory biology classes, either from BIO201 or BIO156. In case you have forgotten any of this, I will add a link to a review of this in one of these classes, and you will find it at the end of the video. But let's go through these just now, briefly, and highlight their relevance to us. So let's start by noticing the fact that water is great for transporting things. So for example, our circulatory system as the blood. A lot of important substances are transported here: gases, oxygen and carbon dioxide, nutrients and energy, waste products, hormones, and so on. And of course, in case of plants, water also plays a crucial role as a transportation media, say for example, to transport nutrients. Let's have a look at the next two. Well, two important of the properties that we know for the water to have are cohesion and adhesion. And sometimes students struggle with separating between these two, so let's have a look at that. Here I have a really nice picture of these two forces acting on these water droplets on a spider web. So let's have a look at what is happening here. What we see here is that these two attractive forces result in a spherical water drop that minimizes the surface area. Cohesive forces hold the sphere together, while adhesive forces keep the drop attached to the web. So cohesion is what keeps the water molecules at attach to each other, and adhesion is what causes water molecules to attach to the surfaces due to their polarity. And I think that I have seen these cartoon drawings being used sometimes to explain this. Or I find this drawing that explains it quite well as well. Okay, let's have a look at the next one. So the surface tension of water is what allows, for example, for the bugs to walk on the surface of water. This results from the cohesive forces between molecules at the surface of the liquid, and it causes the surface of the water to behave like a stretched rubber membrane. Well, water provides also a great heat reservoir. We see an example of this in the climate of the area located by the coast. Seas and lakes absorb heat during the day, preventing a sudden midday spike in a temperature. And then during the night, they slowly release the heat, keeping the nighttime temperatures from dropping really cold. Well, we also know that water can take different forms or phases, if you wish. So I think that you all are familiar with solid phase as ice, liquid water, gaseous form as water vapor. And this then has an effect on the distance between the water molecules. The final one on this listing, which I want us to look at, is the fact that water is a great solvent. So many different things dissolve in water. And for this reason, water is sometimes called as the universal solvent. And the reason why this is important for us in human AMP sense is that water carries many solutes in it. So these are substances that are, for example, dissolved in it. And this, of course, makes water a great transportation media, taking us back to our very first point that I mentioned. It is noteworthy to note that some molecules do not dissolve in water. But really, these are not of a huge interest for us in this context. But the ones that we are really interested here are the solutes that dissolve in water. And when they do so, they form a charge. So some examples are listed here. Notice how all of these carry some sort of a charge. This is important to note here. And note that some of these we find in different body fluids, in blood, in between the cells, and inside the cells too. Let's look at some of the most important ones. So, for example, sodium and potassium. Remember these from our sodium-potassium pumps that were needed to establish a charge in a nerve cell. We also have chloride and bicarbonate. Remember carbon dioxide transport in blood in relevance to this. And of course, hydrogen derivatives such as hydrogen phosphate here. So what I guess that this really tells us is that water and electrolytes these are connected. As one of these will move, the other one will often follow. And here we see some important sites where this exchange happens. And next, I want us to talk about how we get fluids in and how we get rid of it. But I think that the good very first question for us is, how much should you drink in a day? Well, the amount is of course dependent on your body size and activity level and so on. But on average, it would be about 2.5 liters. And of course, we want to get rid of the same amount of fluids. So fluid intake and fluid output must be equal. Now let's have a look at what are the sources of the water intake that we have. Most of the water that we take in is a result of simply drinking. So it is the sensation of thirst that is the driving force for water intake. Smaller amounts of the water intake are also from moist food and a little as a result of the metabolic reactions. If you're unclear as for how does the metabolism result in extra fluid, here is a reminder. The formula of the cellular respiration. So, the process of breaking down carbohydrates, so a sugar molecule, glucose in this case, results in water as a byproduct. So, okay, that is our water intake. Let's look at the water output. Well, the majority of water output is through urine. We do lose some amount of fluids also through processes such as evaporation and sweating, as well as through breathing out water vapor. So for the two first ones, these would be through skin. And for the last one, through respiration. A small amount is also lost through feces, but as you will learn in a module about the digestive system, this is a rather small amount, as majority of the fluids is recovered within the large intestines, and the resulting matters that cannot be digested are rather dry. So, it is not inaccurate at all to say that the driving force for the loss of body fluids is through urination. So, this is driven by a need to pee. And interestingly, if you look at the very last point that I have added to this slide, is that the concentration of the urine, so in other words, how diluted or concentrated it is, well, that is controlled by a process called osmoreceptor ADH mechanism. And what you will find is that there are, depending on a textbook, many different kinds of mechanisms that are given to explain these processes. Some are more complex, step-by-step breakdowns of the processes. Some are more simple, like this seesaw-like explanation of the balance, where the two opposing processes interact to maintain the optimal state. And some explain this as a little more multi-step than this, kind of in between in terms of the complexity of what we have seen before. But I think we can do better than any of this. And to explain this, we end up once again looking at a concept that we keep running into time and time again. And this was, of course, homeostasis. So maintenance of the consistent internal environment. So what we will do, we will review all that you saw before in these diagrams and what you will find in your textbook. But we will do that breaking down in a more manageable chunks and remembering the relationship of these events by using the model that we learned earlier about the homeostatic feedback mechanism loop. And I hope that this is something that looks familiar to you from the past, because this is what we will be using to describe what happens. So, the initial stimulus that we have is that there is too much water. So this would result in blood being too watery. And we detect this with sensors that are osmoreceptors, and these are located at the hypothalamus. And as usual, our control center is in this model the brain, of course. And we act through a mechanism where the effector is the pituitary gland releasing less ADH. This results then as a lesser permeability of the distal convoluted tubule of the nephron. Remember this from our last chapter. And this in turn results in more urine being made, so as an increases excretion. And this, of course, results in us needing to pee, which then in turn brings down the amount of extra water, and we get back to the normal, what is set at the set point for a normal value. So that's the process explained here using this homeostatic mechanism loop model. Now, let's have a look at the opposite situation. So here we have, as a stimulus, too little water in the body. So as a result of our blood, blood is too concentrated, if you wish. Well, this is again detected by the sensors, which are our osmoreceptors at the hypothalamus. So nothing different on this from the previous. And again, the control center will be our brain. But now, as an effector, opposite events take place at the pituitary. This time, more ADH is secreted, which results in more water being taken back at the distal convoluted tubule, which results in less pee being produced, so that we are able to bring down the water amount to the normal as set in the set point. So that's the process explained again using the homeostatic mechanism loop model. Next, I would like to discuss with you the concept of tonicity. And again, this should be something that is a revision from what you know from the past. If not, I have a great classroom video available for you. Just check out the link at the end of this video. So what we will look at here is 3 different situations. We have hypotonic cells, isotonic cells, and hypertonic cells. And we will discuss all of these one by one. But before we do, I want to remind us of this concept of the concentration gradients. So how materials move due to diffusion from higher concentration to lower concentration. This process took place until the concentrations had evened out. Let's see an example of that. So here we have a glass of water with some food coloring added to the left corner of it. So the concentration of the coloring is high at that corner and low at other areas. So what will happen over time due to diffusion? And if you said that the food coloring will spread out until it is in even concentration in all parts of the glass, well, then you were right. So this is an important concept of what we will build on when we look at the tonicity. And I want to start with hypotonic cells. In hypotonic cells, inside of the cell is more salty than the outside of the cell. So what we could do to try to deal with this? Well, the body tries to balance out this difference, and the easiest way to do it is by bringing in more water inside the cell until the concentrations of salts are even on both sides of the cell membrane, right? And this is exactly what happens. So the water is flowing from outside the cell to the inside the cell. And as a result, the cell is growing bigger and swelling and may end up eventually bursting. So hypotonic cells, they end up swelling. You can think of a mnemonic that hypo sounded a little bit like hippo. So these are hippo cells. So big, swollen cells in size. Well, how about isotonic cells? In these cells, the concentration of the salts is the same on both sides of the cell membrane. So there is a balance in place. So, while some water moves into the cell, same amount of water moves out from the cell. So the overall net gain or loss is 0. The cell remains the same size. And again, I have a mnemonic for you. Think of a statement. I'm so perfect. And this means that I am perfect. I don't need to change at all. So I remain the same. And finally, I want to look at hypertonic cells. These cells have the outside of the cell as very salty, while the insides are not salty. So again, we try to balance out this difference, and how it is going to happen is in this one is that water will flow out of the cell, trying to dilute the outsides of the cell. And as a result, this leads to the cell shrinking as it loses water. And eventually, it may even be the case that the cell dries up as a result. Well, Can we think of a mnemonic for that? Of course we can. Think of a person who is hyper. These people have a lot of energy, so they run around a lot. And as a result of running around a lot, you become skinny. So these are our skinny cells. So, just to recap, hypotonic cells were our hippo cells. So they were gaining water and swelling because the inside was more salty than the outside of the cell. And this could lead to even cell bursting. Isotonic cells, they were so perfect. They did not need to change. They were perfect as they are. So there is no net gain or loss of fluid. The cells remain the same since the salt concentration is the same both inside and outside the cell. And finally, our hypertonic cells. These were our hyper cells. So our skinny cell. And they were skinny because the outside was more salty, so the fluid was flowing out from the cell. And as a result, the cells were shrinking and might even end up drying up. I hope that these mnemonics helped you. And finally, if we think of when we might have seen examples of these in real life, it is easy to find the logic in this. Have you ever exercised a lot and been drinking a lot of water because you were sweating a lot too? And afterwards, you get this craving for salt, say for potato chips or French fries. This is your body trying to replace the salts that you lost when sweating. Or you can even die out of drinking too much water within a very short period of time without eliminating these. There was this radio station that held a competition called Hold Your Pee for a Wee when this new gaming console first came out to the market. And someone actually died. So the moral of the story is, I guess, that everyone should study some basic biology classes before coming up with stupid and dangerous competitions. So let's think of one clinical application of what we have just learned. We will have a look at electrolyte exercise drinks, or sometimes these are called recovery drinks. So what we have going on is that after heavy exercise, there is a sweat loss, and we will rather, not surprisingly, get a drink. So now that we have drank a lot of regular water, our body has replaced the lost fluid, but not the salts that were lost with the sweat. So actually, our body fluids are becoming too dilute. So what does our control center do? Well, our osmoreceptors at the hypothalamus sense that as a response, we get a signal to urinate. This is an attempt to balance the fluids to compensate for that loss of salts. But as I'm sure that you realize, this would only lead us to becoming more dehydrated. So not a good outcome at all. So what can we do about this? Well, the solution would be to drink recovery drinks that contain electrolytes. So replace these. And let's have a look at this a little more. Well, these electrolyte drinks serve a twofold purpose. They replace the salts that have been lost, but also help by promoting water uptake by containing important salts for us. I thought that it would be interesting to look at what electrolytes do these different drinks contain. So here we have these. Well, I am not going to advertise one over another one, but you can look at yourself what contains what and how much. This might be important when choosing the recovery drinks. And the last topic that we will need to cover for this chapter is the concept of acids and bases. So our pH regulation and maintenance. And much of this should be reviewed from what you learned in BIO201 or BIO156. Just as a reminder, pH scale ranged from 0 to 14 and the pH 7 was our neutral. And these numbers told us really about the hydrogen ion concentration, with each unit change in the pH scale corresponding to a tenfold change in hydrogen concentration. And this hydrogen that we were looking at, it is produced mainly as a byproduct of metabolism. It is important for us to note that most reactions in the body are affected by pH, so this is carefully maintained within the ideal range in the body. Now let's have a look at some of the normal pH values that we expect to see. Well, the normal blood pH is maintained within 7.35 and 7.45. And if you think about this, this is a very narrow range. So I think that this is quite an impressive accomplishment. And it certainly tells us that maintaining it within this tight range is indicative of the importance of having it so well regulated, as we noted at the very beginning. Another thing to note is that the blood pH is slightly alkaline, or basic if you wish to use that. So that is something to make a mental note about. And next, what I would like to look at the situations where the pH is off from a normal range. What kind of conditions we have in these cases? So if the pH is above acceptable range, we have a condition known as alkalosis. So here the pH is too high. And if the pH is below the normal range, this is known as acidosis. So we have too low pH. So I have a question for you. You might have heard about these diets where you eat based on the food's pH. Can you really change your body pH based on the food that you consume? One example of this would be an alkaline diet, where you eat things that have a pH above 7. Our bodies actually do a great job of keeping our blood pH exactly where it needs to be. Foods likely do not have a significant influence on pH levels. But more alkaline foods like leafy greens, sprouts, avocados, and other powerhouse plant foods provide our bodies with essential vitamins, minerals, and phytochemicals. The acid side of the pH scale also includes healthy foods like tomatoes, whole grains, and beans. Foods that are often considered less health promoting, like animal products and refined carbs, these also fall on the acidic side of the scale and should be eaten in moderation. So I guess that the take-home message is that no, normally your body does keep the pH where it needs to be. So these diets have nothing to do with changing body's pH. Rather, they might help to guide to pick foods that are good for you for other reasons. But your pH is not altered by these diets. So keeping the pH regulated within this range is important. And next we will have a look at 3 mechanisms that serve on this pH maintenance. Let's get to it. So buffers provide us a very fast, almost immediate method of controlling pH. These act through neutralizing the substances that would otherwise change the body pH. So these are chemicals that bind with the acid or base. The next method that we have is through removing carbon dioxide with respiration. This is slower, taking minutes to act. And the mechanism is through breathing regulation center located at the brainstem. This is where the changes in hydrogen concentration are detected, and then as needed, the rate and the depth of breathing is modified. Finally, we have renal regulation through kidneys. This process is very slow, taking hours or even days to act. So the idea is that we urinate out hydrogen ions. And of course, we just had a look at this in our previous chapter about the urinary system. So really, I think that the last thing for us to do is to look at acidosis and alkalosis a little more as a clinical relevance. So here we have some symptoms of the acidosis and alkalosis. Remember that acidosis was to do with too low of a pH. So for example, we see that the nervous system becomes depressed. There can be confusion and even at the extreme case, coma. One example of this would be the uncontrolled diabetes, which may result in a diabetic coma. Then, for the alkalosis, remember that we are now talking about too high pH. Here the nervous system is going on hyper mode, if you wish. And we can see, for example, tingling and even paralysis. And the very final concept that I want to mention just quickly is fluid therapy. So here we administer fluids to the vein. So this is where the term intravenous comes in order to help to maintain blood volume. This can be, for example, in response to a significant blood loss or losing fluids for some other reason. And one thing to note here is that this fluid has to contain electrolytes so that we do not end up accidentally disturbing the salt balance of the body. So some have described these just as very expensive salt water, but really there is more to it. This has to be sterile, the amount of electrolytes in a correct concentration, and so on. So that's it. We have now come to the end of this chapter on fluid, electrolyte, and pH balance. I know that it has been a lot to cover and it has been very physiology oriented, which can be a change from our normal AMP lecture structure. I hope that you have found it useful in appreciating the important work that the urinary system does and to spot these links between various other body systems. Thank you for having joined me for this session.